Hello there. Welcome to DNA Edits. Today's topic is why ligation at lower temperature. That's a topic of our discussion. Joining of two different DNA molecules are very important in the production of recombinant DNA molecules. In vitro reaction or in test tube, ligation happens only at 4 degrees centigrade or 16 or 25. 37 is not preferred. Why? That is what we will discuss today in our lecture. In vitro, the ligation of DNA molecules at 4 degrees centigrade or 16 or 25 degrees centigrade are formed instead of 37 because the high temperature destabilizes the hydrogen bonding of overhangs of the vector and insert. Melting temperature of overhangs are quite lower, hence high temperature destabilizes the hydrogen bonding between the cohesive ends, vector and insert DNA molecule. Whenever ligation is performed in vitro, the hydrogen bond formation between the overhangs is very important to establish the phosphodiester bond. But if you perform this reaction at the 37, the hydrogen bonds which are formed between the overhangs and that get destabilized. So because the overhangs structure is just only four nucleotides. The length of the overhangs are only 4 nucleotides, and the temperature required to destabilize the hydrogen bonds between these 4 nucleotides are quite lower. Hence, the ligation is performed at 4 degrees centigrade or 16 or 25, but not 37 degrees centigrade. This concept I will explain with my own diagram in the next slide. So this is a vector and this is an insert, right? They are digested by the EcoR enzyme and generated the AATT overhangs. AATT overhangs, right? When you ligate them at 37 degrees centigrade, what will happen? The hydrogen bonds formed between these overhangs, are destabilized by this temperature, this thermal energy. The thermal energy at 37 is more than enough to destabilize the hydrogen bonds formed between the overhangs, which are just tetranucleotides. What will happen? This destabilizes the DNA hydrogen bonding. Then T4 DNA ligase do not have any structure to establish the phosphodiester bond between the adjacent nucleotides of the same strand. Hence, if you perform the ligation at 37, the molecules will not ligate. Instead, you have to choose the 25 or 16 or 4 degrees centigrade, depends on your environment. The same thing I'll explain here. When you ligate the vector and insert which are bearing the cohesive complementary ends, right? If you perform the same ligation reaction at 25 or 16 degrees centigrade, what will happen? This 25 or 16 or 4 degrees centigrades are conducive to maintain the hydrogen bond between the overhangs, complementary overhangs of the vector and insert molecules. So the hydrogen bond which is formed between the vector and insert overhangs, they form the structure. That structure is very, very important to establish the phosphodiester bond between the adjacent nucleotides of same strand. Now ligase will act on this structure and establishes the phosphodiester bond between the adjacent nucleotide of the same strand. Now the recombinant DNA molecule is produced. What temperature recombinant molecules are produced? At 25 degrees centigrade or 16 or 4 degrees centigrade, but not at the 37. Hence, most of the times, scientists keep the reaction at 25 or 16 or 4 degrees centigrade, but not at the 37. You just remember. Okay, whenever you perform the ligation reaction, just perform at the 25, 16 or 4, but not at 37. If you don't want a ligation, go for 37 degrees centigrade. Thank you all. If at all you like the concept of explanation, just subscribe for more updates. See you all again with the one more video.